We have two panelists who were not speakers, and they're here to provide reflections based on their own area of expertise and research. The first is Dr. Michelle Lamero. Michelle? Okay, Michelle is at the University of California, Davis. She conducts integrated toxicological and epidemiological studies to better understand the susceptibility of humans to environmental disease, including environmental insults during development and combinations of poor diet and ensuing metabolic disease, and then also focusing on genetic and epigenetic changes that happen with environmental contaminants and how they influence those diseases. So Michelle got a PhD in toxicology from the University of North Carolina in Chapel Hill, and her MPH in Epi while doing her postdoc at Mount Sinai. So Michelle is going to join us for the panel. And the second panelist we have joining the speakers is Dr. Jennifer McPartland. Jennifer is with Environmental Defense Fund. She's been focusing on advancing science, policy, and market solutions to protect human health and the environment from harmful chemical exposure. She's been involved in EDF's engagement with the federal government to advance the new chemical testing procedures and approaches, and currently serves as a member of EPA's Board of Scientific Counselors for Chemical Safety and the Sustainability Committee. Okay, and Dr. McPartland has a PhD in microbiology from University of Chicago. Okay, so if we can have the speakers and the two panelists up here.
So I'm coming at this from a toxicologist hat, if you will. One of the things is the kind of inherent struggle between the order in which we do things. So on one hand, we just heard that it takes about 10 years to come up with a list of 10 good candidates for replacing epoxy resin, and so we would want to know about the toxicity before we came up with that panel of 10, right? You'd have probably a larger panel of toxicants or potential toxicants and candidate new green synthetic chemicals. So in that sense, you'd want something to be relatively higher throughput, which I think makes the Tox 21 type of approach desirable. I mean, the trick with that is it really only represents the first stage in a pathway, and so it can be compensated for or defended against and not end up being maladaptive, right? So if you were really in this push where you needed some answers quickly and you knew that you wouldn't have perfect information and asking quick questions, it wouldn't be a bad idea to screen for the estrogen receptor in one of these assays like Tufts University does in order to determine the estrogenic effects. Unfortunately, there are a lot of aspects of toxicology that are not well represented in Tox 21 type high throughput assays. So for instance, a lot of pharmaceuticals fail to go to full clinical trials because of cardiotoxicity and immunotoxicity. And part of that is because we know that, for instance, the immune is a system, and it's not a cell in a specific dish. And so I think between metabolism, endocrinology, and immunity, it's really difficult to come up with a good way to high throughput those particular outcomes. The other thing is, okay, cardiovascular disease is the #1 killer. What's 2? That's cancer. The key characteristics of carcinogens, if you go through that list of what those properties are, they're not really well represented in Tox 21 or these other high throughput avenues yet. And so I think there's an opportunity for innovation in terms of developing new assays to kind of fill these gaps in some ways. I think in some other ways you'll never get all the way there because you'll be hard-pressed to develop an immune system in a dish anytime soon. And so I think the complementary approaches that we heard about from people today where we're going in vivo and actually, if you want to know if it causes cancer, go into an animal and look for cancer. So thanks.
Yeah, so you hit on some of the remarks I was going to make, but I think just taking a step back, in hearing both of these presentations and then also just kind of being in this space for a while around predictive toxicology approaches, I think we can all agree that they provide really valuable information, in particular in the data poor environment in which we often find ourselves, where there's either insufficient data or no data whatsoever. And I think we've heard that from presenters today, and that's something that John pointed out, Dr. Bucher pointed out during his presentation on MCHM. And I think, speaking from a pragmatic standpoint, given the number of chemicals that are lacking data, and all the toxicological endpoints that we would like to have information on for a particular chemical, it's probably unrealistic that we could test sufficiently using only laboratory animals. That being said, I think that there is a healthy spectrum of perspectives on the quote-unquote readiness of predictive toxicity testing to understand what potential hazards and risks there may be of various chemicals. And so, and that really centers on, I think, a lot of what Michelle was just saying, which is the extent to which these methods and tools can accurately reflect whole human biology, where there are a lot of dynamic processes at play, both within cells, between cells, and at the tissue level. And if you think about early life biology, there are a tremendous amount of spatially and temporally dynamic processes going on. And so when you think about trying to capture all of that in reductionist approaches, it can, it's scientifically almost overwhelming a little bit. But I do think that we're getting there, and I also think that we don't have only one predictive tool in the tool chest. We have an array of predictive methods that we are starting to consider. And so I think what we can hope for is that using all the tools that we have in the tool chest, whether it's high throughput screening or transcriptomics or alternative model organisms like zebrafish and C. elegans, we hopefully will balance out the strengths and weaknesses of all of these things and get data that is meaningful and ideally actionable. And I think the last thing I'd note on the predictive tox aspects of things is that I think it's really important to be very transparent about the tools and the methods, what they can measure and what they can't measure, and what the uncertainties are. And I also think it's really important to continuously kind of advance the methods by bringing what we're learning in basic research around disease, etiology, and pathology, and bring it back into the testing system so that we know that our new testing tools are accurately reflect our current understanding of human biology and health effects. I have some questions for the presenters, but I will wait until the moderator says I can ask them.
So they need to mic me. Really? Usually accused of being rather loud. So I think that what we just heard points to the elephant in the living room, which is we never really have enough data on toxicology and adverse health outcomes for chemicals in commerce, and that includes pesticides as well, really, because the guideline studies don't ask all of the questions that need to be asked about a chemical's interaction with biological systems. So it's really difficult for people trying to do the right thing and replace a chemical that everyone's worried about with something that is going to be safer. So I think the emphasis has to be on safer within the confines of what we can test and what we can know. So before you ask your questions, I'm going to ask these folks if they have any comments on what has just been said. So Dr. Bucher?
Well, yes. So I appreciate the comments from both of you, and I agree with almost all that's been said. So the Tox 21 program is, you've only seen a part of it here today. The full program has a sort of a tiered approach to generating data in vitro, primarily in human cells, and then moving on into what we call orthogonal assays, which would be a second technology that would either confirm or deny the first indications out of that first technology. And then zebrafish and C. elegans and other alternative species were considered part of that whole process of validation of the outcomes. And I just lost what I was going to say, the next part of it. But I think that, when the Toxicology in the 21st Century NRC report came out, there was a prediction that it would be 25 years really before this really hit the ground and you could really run with it. But I think that the technologies have actually advanced more rapidly than we anticipated. The costs of sequencing have gone down. And the other thing that I do remember now that I was going to say is that the building more complex models that are somewhere between cells and animals is really advancing rapidly. The tissues on a chip, primarily because of the large amounts of money that NIH and the Defense Department have put into this effort, are creating lungs, kidneys, livers, and you're beginning to see the processes where they can make those things work together. So these are not going to be high throughput screens, but they're going to be screens that will not require animals and that can be done in fairly rapid order, and you can begin to get at the complicated biologies, and the immune system on a chip is going to come. It's going to be there. So we'll get there.
Dr. Mayer, would you like to comment on anything so far?
Sure. H.R. Mencken has a quote that some of you may have heard: for every complex problem, there is an answer that is clear, simple, and wrong. And I think that biology is a complex problem, and that you have to be very careful and thoughtful in how you interpret data that are biological in nature. And that all of the data that was just talked about may be inductive in that it is hypothesis generating, or I'm suggesting that it may be. And that you actually have to generate a hypothesis and deductively prove that hypothesis, otherwise you're wrong. And plausibility doesn't mean it's necessarily true.
So, Jennifer, would you like to ask the questions you had of the earlier speakers?
I actually had a question for both, but I'll start with John. In the BPA Consortium study, right, where there seem to be like over a dozen investigators that are like examining different biological aspects of BPA. Do you envision that the data that's generated through the consortium could help to provide some direction as to what should be examined in BPA alternatives from at a molecular level?
Well, the intent really of that program was not to study BPA. The intent of that program was really to look at what's required from a guidelines study standpoint and the designs and the kind of information that's being generated, and compare that with the academic, the ability of academics to generate information that seemed to be at odds with respect to BPA. So it was a perfect candidate. It's been studied ad nauseum by thousands of papers have been generated, and yet there's this clear divide between what one would expect about the human responses at very, very low doses from academic studies versus the regulatory guideline studies. So it was to try to point out whether which one is closer to the truth in terms of being predictive for the kind of things that we need to take account of. I don't think that the BPA alternatives are going to have to go through that kind of an exhaustive approach at all. And I think almost no chemicals should go through that kind of an exhaustive process, because the purpose of that really was to see whether we need to tweak their guideline studies to make them more sophisticated and more sensitive.
Yeah, no, I was thinking it more in terms of that there are molecular insights that come out as a result of the study. Would those be the same molecular insights then that we may want to examine with BPA alternatives?
I think that's a fair assumption, sure. And a short answer.
Okay. Are you done?
No.
Before
Dr.
you have a question?
Beaker. So I noticed in one of your slides, you had it was a chart, and then along the right-hand side of the slide, you had it was the ToxPy slide, right?
Okay.
And I noticed there that you had estrogen receptor agonism, right? And then you had full receptor and partial receptor.
Mm-hmm.
And for some of the compounds, there was over a magnitude order of difference in the level of agonism between having the full receptor and having the partial receptor. What do you think that that's about? But that wasn't the case for all of them. In other instances, you saw them much closer.
Well, since we're not talking about BPA toxicity, I mean, I really can't answer that question. The reason we developed the full receptor assay was because we weren't getting the kind of responses that we would have expected to see with the partial receptor. And it has to do with docking and binding of those chemicals with the receptors in ways that I'm not sure we have a full answer. But it just adds to the complexity of the issues. That's all I can say.
I was thinking if you had only chosen one of those estrogen receptor, estrogen agonism assays, you could be getting very different results.
Yes, and that's one of the reasons that we have the orthogonal assays built into the program so that we can verify the responses.
So I have a quick question, and then can we go to the audience? So my question is for Dr. Mayer. So I totally understand why you guys focused on whether or not you could find a bisphenol that did not have estrogenicity. And I'm really impressed with the amount of work that you guys did to find one and to go through the motions of showing it was not estrogenic. Was there additional work done to look at other types of endpoints or other mechanisms by which the monomer you guys came up with that looks really cool and probably works has other types of adverse effects?
Sure, you have another hour that I can talk about that. I tried to condense what we were saying. We've looked at thyroid, we've looked at androgen, anti-androgen, all of the endocrine sort of array, if you will. So, systemic toxicity for all these monomers, and yes.
Yeah.
The short answer is yes, and it's that one table I put up there that had the columns on it with all the different monomers. It shows all of that.
Okay.
So absolutely.
All right. So let's go to questions in the audience.
So I could have listened to Mark and Marisol for a very long time. That was a fabulous presentation. And I was wondering if you would just speak a little bit more about your, I can't remember, I think it's called Valspar Safe, like what the key components are of the substantiation for safety that Valspar now uses to evaluate safety of the materials it creates, and whether or not you think it could be a model for the rest of the industry. And I mean, not being at Valspar anymore.
Well,
Can you still answer that?
Yeah, you're talking about safety by design.
Yeah, that's
right?
what I meant.
Okay, so safety by design. The one thing I didn't want to do is sound like I was pitching anything, because it is kind of a trademarkable sort of statement to make. Safety by design is nothing more or less than absence of evidence. And to do that, it would be nice to have a paradigm like TypeHead or green screen where you can check the boxes as you go down. But like I just said about complex problems, H.R. Mencken's quote on complex problems, the safety by design, if you want to translate it, it means collaboration with people that are going to tell you you're wrong. It's not a checklist, and it would be nice if we could do that. But Janet, I don't think it is a checklist. It requires input from people like you, people from Tufts, and somehow we've got to figure out a way to communicate between ourselves to get those kinds of challenges, to find out what is safe. So that's part of the design. Part of the design is to get the people that disagree with you to weigh in on what you're doing. That's the best I can say. I'd love a checklist, but we don't have one.
Say something?
Sure.
I just, because I didn't say this in my opening remarks, I did want to say, like, the degree of transparency that that Valspar has undertaken with this alternative molecule is really laudable, and I don't think that I've seen anything like that in the time that I've been at EDF. And speaking from an advocate's perspective, I would love to see more of that. And I wonder if, having gone through this experience, would you guys do it again? Or not you guys, but Valspar?
Well, the story's not written yet. And so the answer is, today, looking back in the rearview mirror, and by the way, a lot of this is, there's a lot of chaos in the rearview mirror, but when you look in the rearview mirror, it all fits together like you were really smart, right? but we weren't that smart. A lot of it was saying, well, that worked, that didn't work. But when you line it up behind you, you go, well, that was cool. Okay, so honestly, to date, everything we would do over again. And speaking for me personally, and I would guess speaking for Tom Allen, we would do it again. And the appreciation and the recognition that we've had from all of you is really encouraging for us. And it's always encouraging to be a leader. But like I said, the story's not written yet. We'll see. We'll see. Ask me in a year, ask Tom Allen in a year, and then we'll see where we end up. But yeah, so far so good.
Tom Lent, Healthy Building Network. If I can just follow up on that a little bit, again with applaud it, and also the question, because we, Healthy Building Network, we're pushing for transparency across a wide range of manufacturers, and we find the formulated products industry to be the hardest to work with, and and they for the exact opposite reasons that you're explaining, because they think that there's no way that they can have their intellectual property protections in place sufficiently, and generally tend to point overseas. How are you guys different? How are you able to go opposite this trend that we've seen of formulated products manufacturers having no trust that they can have their intellectual property protections in place sufficiently to go as transparent as you have?
In some ways, the question you're asking,
or are they bullshitting me?
Can be, well, no. I mean, legitimately, formulation is a bit different than this. What we did at Valspar is more like a pharmaceutical development project, if you will. It's not a pot full of stuff that you empirically put together that works really good and you go patent it. I understand the dilemma that they face. What we did, on the other hand, was more like a pharmaceutical project that says we've got several protections built into that. Of course, you have the patent and intellectual property protections, but you also, as much criticism as regulation draws, that regulation stuff is expensive, it's time-consuming, and it buys you, essentially it buys you time. It's like something going off patent, right, for a drug. They get about 10 years of useful life out of that drug. And then hopefully by that time, they're on to something new. So that's the idea of innovation, is you buy yourself some time, you get the patent protection, you get the regulatory protection, and then you go like crazy after that next innovation so that everybody's like trying to catch up with you all the time. And I think that's the way this works. Formulators, I can't answer the question because I'm not one, I've never been one, but I understand their problem. And I don't have an answer for you, sorry.
Yeah. Yeah, Mark, kind of a follow-on to that, Tony Kingsbury over here. What's the size of the market in general terms for the can coating industry? I mean, I assume that played into Valspar's investment, and I assume it, I mean, based on all the testing that you showed, do you have any sense of the investment that, kind of put into this product development?
Are you asking for a number? No, I'm not sure.
Sure.
Well, drugs nowadays are costing $1 billion. Okay, we're probably in the 10% of that space. Okay, just an idea, and I can't give you any better number than that. But it's very expensive to do this. Okay, so you have to recover that cost somehow. But the other question you're asking is the supply chain, sort of, how does it work on both sides of you? And I mean, there's the top raw material guys, and then there's the downstream guys, which if you want to go all the way downstream, you are at the retailer level. So your Walmarts and Costco's and those guys, those are at one end, and at the other end of the raw material suppliers. And if you back up from wholesalers or retailers, you get to brand owners, you get to can makers, you get to Valspar, then you get upstream for the chemical supplies. I have to say that is the most onerous problem that we never imagined how difficult that was going to be. Because you're stuck in the middle. You are literally like the cat stretched across the bathtub, right? You're looking down and you're going like, man, I got people pulling me in both directions. Now, this is Mark talking. This is not Valspar talking. This is from my experience, like my cat's experience, right? And the trick is not to fall in, because you're going to have forces pulling you both directions. Your upstream people don't want to share their intellectual property with you, even though you're sharing it with the world. And then the brand owners have a lot at stake, right? The brand owners have a lot at stake because they're front-facing. They're looking at client. They're looking at customers. They're looking at all of you. And then the retailers and the wholesalers have the same problem. So there's a lot of opposing forces. I suppose in a way, you know, we're taking fire from both directions. We, Valspar, they, Valspar. And I don't have a clear answer for you, but other than to say, if you're going to make change, you've got to have the guts to stand up and do it and take the fire. And I don't really have a clear answer for you, but it's the cat across the bathtub problem.
Mark's an inch taller now. Did you say from when you started?
Yeah. I don't know. I didn't really answer your question. I know I didn't, but I don't know how to answer your question. I guess is why I'm rambling.
So I'm actually working in the waste industry. This is a little bit different than what you've been talking, but it's related in the sense that how you develop an alternative that is safer. I talked to so many different recyclers. One of the major problems is they have no idea what the manufacturer put in. And if that's quote, quote, recyclable, how would that be separated? Is any food packaging company in the future think about the product stewardship or extensive producer responsibility in the sense that when they develop a safer alternative, they know all the chemistry in and out and how to separate them. Because rely on cradle to the grave, we always stuck with the problem. Just 6 months ago, I actually got 2 couches. The customer was really conscious that this is a brominated flame retardant. This is toxic to me. And I told them, there's no way I can help you to get rid of us through a hazardous waste except landfill. Mm-hmm. So this is where the things start coming back too, because recyclers, especially metal recyclers, they always complain about plastic material in there. So, you know, when you develop something new or novel, think about the waste industry.
Yeah, sure. So you've now stretched the space even further. Well,
exactly.
For cans, that's an easy one because cans are really cool because they're 100% recyclable indefinitely. Do you have any waste? Yes, you have waste, but most of it gets incinerated in the smelting process.
Exactly.
So then it's just managing the air emissions if you want to look at it at that level. Okay, so every industry has its like, oh gosh, we're lucky that we don't have to deal with that. Well, the can makers, the can world, we're lucky we don't have to deal with that. But I just had a thought that had to do with the formulation question that applies to what you're asking, is how do you know what's in this stuff, regardless of whether you're trying to dispose of it or you're trying to put food in it. I would argue that, and again, this is Mark talking, I would argue that all of these formulations, all of these products, all of these materials have what I would call sentinel components. Components in them that if you knew that component was in there, it wouldn't allow you to go out and make it. But it sure as heck would allow you to determine the hazard or risk of that material. So I think it is maybe a good call for formulators and manufacturers like waterproof rainwear to say, okay, there's a lot of stuff in there we're not going to tell you. But we can tell you about the 3 molecules that are of concern. That's only fair. Now, you have to trust them, right? But then again, if you have peer review, even if it's under confidentiality, you can have someone say, these are the molecules you should let people know about. It can be done. But go for those 3 sentinel molecules, because in a can coating, I can tell you there's a lot of other stuff that goes in there. But all that other stuff is like waxes and things, lubricants and stuff like that really you're not terribly concerned about. Nobody would be. So ask that question. What are your 3 or 5 or whatever should we be worried about?
But the trouble is most of those PR or even safety guys, they only tell you, look for the SDS. And the SDS has nothing except proprietary ingredients.
I know.
I think you're pointing to an issue that we're unfortunately all too aware of, is that we don't have a nice little paper trail where all the chemicals go in our technological society.
My question is for Drs. McPartland and Lamerrill. I'm Meg Schwarzman with the Berkeley Center for Green Chemistry, and also actually probably Dr. Bucher a little bit. I wanted to hear something about your wish list for endpoints. If Valspar had unlimited, or anybody creating a new can coating, can lining, had unlimited resources. I know that the testing that we currently have available to us doesn't adequately reflect biological complexity and our understanding of the biological systems we're interacting with. But what would you put on your wish list from what's available?
I could start. Yeah. I think an immune system would be a good one. The cardiovascular and the characteristics of carcinogens that I mentioned previously. I also think for instance, the EPA definition of an endocrine disruptor basically is estrogen, androgen, and thyroid, and the endocrine system is quite complex beyond that. Insulin, for instance, is underlying the etiology of obesity and diabetes, which are highly prevalent diseases in our country, and we don't really have any assays that are routine to do it in there. I don't recall that you have an insulin receptor assay in your receptor list. But for instance, in our lab, we do a kind of medium throughput cell-based screen where we look at insulin action by stimulating adipocytes with insulin and seeing if the chemical in question modulates the ability of the cells to take up glucose in response to insulin. So that's kind of more of a physiological-based test, in the same way that at Tufts they have the e-screen, which is driving proliferation with a known underlying pathway. So I think those kind of intermediate things where they're a little more anchored to physiology, like broad speaking, is a nice strategy where you're kind of saying, well, it's not quite as simplified as a receptor, but it still allows some level of screening before you get into a whole organism. I also think it would be really nice if, for instance, breast cancer is always a big scare, it seems like, and a lot of these, what's the adverse outcome that we're trying to protect ourselves from? And a lot of the rodent models that have found toxicants associated with increasing rodent breast cancer risk also modulate the growth and development of the pubertal mammary gland, or the breast of the rodent, which we heard a little bit about earlier from the speaker. But the assay's a bit limited in the way they're doing it, because they're looking at juveniles in a single time point. And what they found in those other studies is it's not always the exact same kind of perturbation of puberty. Sometimes it's a bit of a delay and then a rapid compensatory growth, and it looks fine at the end. Sometimes it seems like it never fully matures. Sometimes it seems to be more growth early on. And so there's all these things where it just seems like, well, maybe there's any changes adverse. But I think you'd have a lot better of a picture on it if you had 2 time points, and you could also see that a number of these are more kind of dangerous during the prenatal period. And so you can have a pretty efficient design where you expose a rodent while it's pregnant, and then the offspring can be enrolled in different time points of the assessment so that you have the litter mate controls. And so there's little things like that I think you could do to add value and have a physiological, developing, immune, endocrine system. That would be a relatively efficient, short time course in terms of experimental costs. So.
I think it's really, really critical to understand when you have a chemical that's got widespread exposure, that you need to be looking at developmental outcomes. Jennifer.
Yeah, well, I was going to say developmental outcomes, the potential chronic effects. I know that we had focused a lot on the reproductive effects of BPA. They're not supposed to be talking about BPA, but BPA is in the title of the meeting, so it's really interesting. There also have been some human epidemiological studies that probably folks here are familiar with that seem to show that there's some behavioral impacts potentially of early life exposures to BPA. So I don't know that we understand molecularly exactly what's driving those effects, but again, it's another, I think, illustration of a developmental effect. And so those chronic long-term effects would be on my wish list. CMRs, you know.
I remember that estrogen's involved in brain development, and so that there is a logical reason to be looking at neurodevelopmental toxicity with estrogenic compounds or anti-estrogenic compounds. So I have two questions that came up from over the web. It says, Mark, you tested TMB-PF for mammary gland endpoints and endocrine disruption, even though the tests aren't required by FDA's food contact substance regulations. Why?
That's a question that Dr. Maffini can answer better than I, because she's part of that magic. So please, can you give her a microphone?
FDA has recommendations for what should be tested, and those recommendations are usually grounded on a set of prescribed tests based on the level of estimated level of exposure. The great majority of the time, the level of exposure that is estimated by a manufacturer doesn't go beyond a genotoxicity study. Now, in this case, the hypothesis was to test whether or not there was estrogenic activity, and to be logged into just guidance type studies like the eutrophic study that has been questioned by its lack of sensitivity, and also having other endpoints of higher sensitivity to estrogenic activity or anti-estrogenic activity, like the mammary gland has been shown by many different laboratories be one of those organs that is very sensitive. Valspar was willing to get out of what the regulators would ask for. And I think that was a very important point, because you really need to figure out what is your most sensitive measurement to be able to determine if something is working or is not working. Similarly, with the e-screen is also not in the books. It's not a standard test or even a screening for chemicals, and not even perhaps in the radar for final products. So I think the fact that they were not intimidated by trying new things and really making sure they were driven by the fact that they want to make sure the product was safe or didn't have this particular activity, it was very interesting.
While we're on the subject of toxicity testing, I think it would be kind of fun if you gave TMB-PF to John and had him run it through all those assays, since you folks have developed a number of toxicological testing results. You can see how it compares to the Tox21 ToxCast type assays.
Of course, and Aldrich has some on their shelf. Very little, very small amount, but enough for your high throughput. So yeah, by all means, you absolutely should do that.
So there's another question from our web audience. What has been the reaction from other companies to what Valspar has done in terms of the model in this development and its transparency?
So read that again, please.
Yeah, I'm not sure I'm reading everybody's writing. What has been the reaction from other companies to what Valspar has done? Yeah.
Oh, other companies. Well, we were just talking about the supply chain and the universal sort of the huge spectrum of that. Other companies, I wonder if they mean competitors or they mean brand owners, or I'm not sure how I can answer that. I think I touched on some of the concerns that maybe Valspar's customers have, and they're legitimate business concerns. I would say we haven't heard a lot from anybody but the brand owners. The brand owners, for the most part, and when this is globally, have been incredibly supportive, and they have had a lot to say about what they wanted to see in these assays and what they wanted to do. So I think if you want to talk about brand owners, we got a lot of input and a lot of very valuable information from them.
So I have another question coming in from the web. And they're asking with TMBPF, if I got it right, as you move forward, what are the next steps for incorporating that into can linings and into production? So they're asking, are you being asked to do other targeted testing other than endocrine activity by regulatory agencies? Or?
Yeah, I alluded to that briefly, and we just there wasn't enough time. But you look at all the reasonable endpoints for low exposures. I mean, you wouldn't look at like 100 milligrams per kilogram toxicity because nobody's ever going to be anywhere near there. But you would look at immunogenicity, you would look at endocrine activity, you would look at, I don't know, maybe developmental, I don't know. It depends on if you can actually justify a low dose for developmental toxicity. The answer is those have all been looked at. Go to the website, read the hundreds of thousands of pages of data in there if you really want to answer that question. It's all there. Or call me, and I'll see if it's okay with Valspar, and then I'll share it with you.
Pancho.
I was told somebody else
Oh,
is
sorry.
ahead of me.
Hi, I'm Catherine Alcantara with the Center for Environmental Health and the Change Coalition. I had a question for Mark. Given that now everything's out there and you have this product, I'm wondering what is the availability and what are your projections for the transition of this product? I know you're not at Valspar anymore. I don't know how much you can speak to that, but given sort of the regulatory climate here in California and the consumer demand for non-BPA lined products, I'm wondering what your expectations are for that transition.
It is commercialized. It is commercially available for some specific can uses. A lot of the answer to your question is going to depend on Valspar's customers and Valspar's business partners as to how that looks and how that's done. But it's there, and how fast it moves is going to be really no longer a matter of science. It's now a matter of business decisions. And so I can't address that any better than that. It'll be different in every location from Europe to the rest of the United States and then where we are now, which seems to be a different paradigm. So,
Dr. Moran.
Punch is fine. Francisco Moran from OIHA. I like your vision of a simple answer for a complex problem. Thinking in physiology, an endocrine an active hormone has to be free to be active. Have you ever considered into can lining problem to add something that will block that leaking from the polymer into the food? You know, thinking in physiology you have binding protein, and in the laboratory you have charcoal to separate bound and free. Is that a possibility?
So I think Dr. Lekine addressed that, or no, actually it was Dr. Bradley who addressed that. I'm getting tired, sorry. That you really can't, even glass leaks stuff, okay? I don't think there's anything you can do that would eliminate migration short of pushing it up on the synthesis scheme, which is what we did with the bisphenol. It's up high on the synthesis scheme, so by the time you finally get to the polymer, it's all gone.
Yeah. See, I'm not thinking on changing the molecule, so leaving bisphenol A there and add something that it will block the leakage to the food, so something that will precipitate it, for instance.
Like a film, like a film. Okay, so yeah.
That there will be a protein, so it will be probably a good actually for
in principle, you could do that, and I would love to hear your ideas on it. But keep in mind, cost becomes really, really important when you're doing millions and millions of cans. If you add, a mil of cost to that can, all of a sudden you've changed your cost structure. So that's probably the biggest concern. Theoretically, you could do it, but it would be nice not to have to do that. But yeah, I'd love to hear your ideas on it too. I'm sure Valspar would as well.
We have to move on, I'm sorry to say. So thank you very much. Thank you to our panelists.